It's very nice. We have 10 presentations, several women presenting, as Hilda noticed, which is really nice. So, so I'm going to be the host for this session, and I'll be not very strict, but I will let you know when you are 4 minutes into the talk, okay? So David is going to start, so please.
Okay, thank you very much. Well, as you may know, my name is David. I work at IFISC and Palma de Mallorca in Spain, and my supervisors are Tobias Gala and Raúl Toral. And my work is basically anticipated synchronization, and I am doing some things in a deterministic kind of fashion using distributed delays. And I am also interested in stochastic individual-based models. So let me first explain you a little bit about anticipated synchronization, in case you are not familiar with. So of course, I mean, this is a Coulomb synchronization, and anticipated synchronization is a kind of synchronization, but it's not that the two systems are doing, well, typically we can, let's imagine that we only have two systems. So it's not that the two systems are doing the same movement at the same time, it's that one system is anticipating, so it's sort of forecasting the state of the other system. And this is pretty interesting because it works for linear system, which is sort of boring in a sense, but it can also work for chaotic system, like this example that you have here for the Rosler system, and it can also work for excitable system. So one configuration to get this, which is the main one that I'm using, is not the only one, but it's the main one, is to have, like you can see on the top right, the top left, sorry, you create two identical copies of the system, same parameters, same everything, and one of these copies is left unchanged, so you do not modify its dynamics. And then you create a second copy, and this copy is coupled to the state of the first copy, and it's also coupled to itself, but with a given delay. So the equations are the one that you see on the bottom part. And of course, I mean, this for me at the beginning, it was pretty surprising or even not so trivial, because I was like, okay, you have two copies of the system, and you are just you are not modifying the first one, but you are telling me that the second one is able to forecast the other. So it's kind of confusing at the beginning, or at least it was for me. But I mean, since now you are familiar with synchronization and so on, you can easily see that you can get the synchronization manifold, and you can see that one solution of this system is just that the thing that you have in a box in the bottom, that you have that the driver, the y variable, is anticipating the x variable. So this is pretty nice. Of course, then there are some limits. This is a solution, but it's not necessarily a stable solution. And this has been studied since the early 2000s. So there is already a lot of things here, but what I am interested in doing is, for instance, going to a distributed delay configuration. So before we have studied, well, a lot of people have studied this configuration with just a single delay for chaotic system, excitable system, spatially distributed systems. But what I want to do is to say, okay, what if we have, for instance, two delays? So on top, you can see, what if we have two delays lines, which I mean, it could happen, no? It could be that your system, for some reason, have sort of two delayed self-loops with different delay lines. It could happen. Or it could also be that you have a distributed delay. Like, I don't know, maybe there is some uncertainty in your delay, and in this case, you will have, effectively, you will have a distributed delay equation. Or maybe, I don't know, it will even be like, this will even be a first step in order to study system in which the delay is itself a random variable. So this is one thing that I'm interested in. I'm also interested in using this knowledge about the deterministic dynamical system into other disciplines as well, and also about synchronization in general. And as I have already also mentioned, other idea that I'm currently working on is like, okay, we have understood sort of anticipated synchronization in these deterministic models, but for some models, I can build a stochastic individual-based model, which is equivalent. And for instance, I don't know, if in this model we have a limit cycle, in this model we will have a noisy limit cycle. And we can, for sure, if there is anticipated synchronization in the deterministic model, there should be anticipated synchronization here. But also the interesting question for me is like, there is some properties here that are not there. For instance, you can have noise-induced oscillations here. So can we find anticipated synchronization in that case? And this is still sort of an open question that I am interested in attaching at some point. And okay, I will finish. And I mean, I'm also interested in general in stochastic individual-based model, and that's all I wanted to mention. Thank you.
Thank you, that's very nice. Okay, Gilles? Okay, I think you can choose one of the two.
Thank you very much. Okay.
Excuse me.
F5. Okay. Thank you very much. My name is Gilles Koudavoke. I come from Benin. Benin is a country in West Africa. So my institute is Institut de Mathématiques et de Sciences Physiques, in English, Institute of Mathematics and Science and Physical Science. So our main language is French, so I ask you to excuse my high quality English. So my laboratory is the laboratory of fluid mechanics, nonlinear dynamic, and modeling of biological systems. We work on mathematical modeling of micro-electromechanical systems. So there is a chip, one example in the micro system that we study in case of my thesis. There is a it's not working. Yeah. Okay. Okay. Okay, yeah. There is the mechanic part and all other the electric part. So we modeling mathematically the system, and we plot here is the variation, a behavior of the microbeam when we vary, we change the intensity of current. The intensity of current. So that is a bifurcation diagram. Here, the Lyapunov exponent to confirm the diagram of bifurcation. So when we choose some value here, we can get this comportment of our microbeam, and when we choose one value in this domain, we can get this comportment. So my work is on on MEMS. We study we modeling mathematically this micro-electromechanical system. Our goal is to clean all possibility to get chaos in the system. So we will remodeling this system to get the periodic oscillation on all time. The goal is to build a hormonal micropump, so that we don't get it now. We don't work in synchronization, but we think we will use it on another time. Thank you. I finish.
Thank you. Okay, next one is Michael. Can you find your talk here? Yeah, there you go. Yeah. Yeah.
Okay, hello. My name is Michael Luckton. I'm a sociologist, and you might wonder what I'm doing here. I'm not a physicist, and, well, I wonder that myself a little bit. But how I got here is my methodology got me here from my doctoral studies, which was at the Open University of Catalonia. And it's a type of methodology for qualitative work where you go into the field, try and keep an open mind, and then you go and you find your theory after you're in there. And you look and see what's happening in the field, and you go to the theory, you go back to the field, and you try and find things that explain it. And so I want to introduce to you four key terms that I found in my field work. And these are terms that I got from my informants, that I found from the people that I studied. Well, what am I doing here? I'm hoping that you can help me and that maybe I can help you, because you know a lot about how to make models and you've studied a lot about synchronization and how it appears in the field. And I know a lot about synchronization because I've lived it, and that was the sort of research that I did. I didn't have to look at the bees and try and guess what they're doing to synchronize. If I wanted to know what was happening, well, my informants, they told me that they synchronized. I didn't have to make up a model and try and decide what was happening. Okay, so, wow. Yeah, so I studied a type of, a community in Barcelona who played a type of music from where I'm from in the Appalachian Mountains. This type of music, it has a type of drive, is what my informants kept telling me. It's a type of way that you have to play sort of in front of the beat. It's inexplicable. It's something that even my informants can't really tell me what it means. It's a type of music. It's a rhythm that you have and that you have to learn face to face. And so it's that face-to-face synchronization that is the key to this sort of research. The more that you synchronize with someone, which happens when you play music with, the deeper your level of bluegrass will get. And people do that by forming these sort of associations. So I just want to tell you real quick about what happened in my research. Well, I would go to these jams that would happen twice a month, and I would play music with people. I would synchronize with them, and then I would do interviews with them for 5-year ethnography. And what I could understand is that synchronization is something that happens when we make music together, and that in order to understand bluegrass, in order to play it, in order to increase your level of bluegrass, you have to learn face-to-face with another person. So the big problem that we have in Barcelona is that because of this face-to-face issue, the expertise tends to concentrate geographically. And this music, it's from a place called the Appalachian Mountains, which is where I'm from. So the musicians in Barcelona, they want to increase their level of bluegrass. They want to learn that drive, but there's no experts around in order to teach them to have that face-to-face interaction with. So what they do is they form these sort of associations. And I try and talk about these fractal associations in my poster, but I want to move really quickly. And they form these associations to help bring experts from the Appalachian Mountains here to Barcelona, so that they can get in these jams and synchronize with the musicians who are there, thereby sort of increasing the local level of expertise and teaching them this sort of drive. So I think that we can all agree that humans are oscillators, that, well, maybe we can't, but I think that it's pretty obvious that we have a heartbeat, and that in order for someone to be alive, to be a human, they have to have a heartbeat, and the definition of not being alive is to not have a heartbeat. And that's a type of oscillation. What's, I think, a little bit more problematic, and something that I'm trying to figure out, is whether these associations, these groups of oscillators together, can themselves be an oscillator. That one, I think, is a little bit harder to see, but I did find that in my research. And sometime during the poster presentation, or when I give a longer presentation, I'd like to be able to tell you about how these associations form sort of nested inside of each other, and it's through these associations that the oscillators are able to bring the master oscillator Well, the person who's got the drive, who's got the deeper level of that rhythm, how they're able to bring them to a local community and increase the level of drive in that local community. And when that happens, you can even get here where there's like a trickle-down effect. So here we've got these different types of associations. You've got your sort of local, which was my community in Barcelona, a regional. Here's the European Bluegrass Music Association, and a global. And when you have a local association, you'll try and have a event that brings people from the larger associations and more experienced associations in. And so in this sort of drawing right here, we can see these people, they normally synchronize with someone closer to the center, closer to the global or the regional level. And when they come here and they entrain and synchronize with the local oscillators, it increases the level. But it's not only the local oscillators in there, but even people on the outside who will entrain with those local oscillators, their drive also increases. So I'm sure this could be explained a little bit more fleshed out in my talk, or if you'd like to come see me at the poster. But luckily, I do have some people helping me out at the University of Barcelona, and thank you, Albert, for getting that worked out with Conrad Perez, and he's got a student who's maybe helping me figure out some sort of modified Kuro-Mata model to help me describe what's going on with this. But if anyone has any other ideas, if you think maybe it could be applied to your fish or whatever it could be, please, I got my email right there, and I've got a QR code that you can look at my LinkedIn page, too. Okay, thank you. Okay, yeah, I will. I'll come see you, and if you could give me his information. Yeah, well, that's why I'm here, even though sometimes I don't know why I'm here. Thank you.
Is it just presenting? Yes. Okay, can I go?
Yes, please.
Okay. Hi, everybody. I'm Amalia. I figure everybody here sort of already knows me. Wait. Let's accept the cookies. Okay, we have the cookies. Okay, so first of all, I'm a student at the Universidade Federal do Rio Grande do Sul. It's the southernmost state in Brazil. And I should start by saying that I'm still in undergrad, so I'm an undergrad student, and this is one of my research projects where we're looking at microswimmers, or paramecia, which are protozoans, and we're looking at sort of how they move. But before that, I want to do some self-advertisement and say that actually this is my main research project, and we're looking at climate extremes in Brazil, and we've actually gotten some results already. So if anybody's curious about that, well, our results are quite obvious. Extreme events in maximum temperature are increasing, it's getting hotter. But if anybody wants to talk about that with me, please come and talk to me. But coming back to the tracking. So what we're doing is actually quite similar in a very experimental level to what Carmen is doing with her fish. So we are looking at these protozoans in a microscope, we're filming them, and we're tracking them to see how they move. And so already doing this is a bit of a challenge because, well, when we look at the experimental data, we start with settings with a lot of dirt and a lot of paramecia, which are coupled together, and it's very hard to get anything from this because we can barely track them when they're very lonely. If they are in groups like this, it's almost impossible, especially with the dirt. So the first thing we do to track them is we confine them so we can see their full trajectories, and we clean the water, and we get only a few paramecia. And you see this is already much better, but there's a lot of shadows. So we end up with an algorithm to take out the background. And so this is our final configuration to enable us to do the tracking. So this is all to get there to that video that Kanmin showed of the fish. This is all that we have to do just to get there. And my part is mostly computational and figuring out what's best for the tracking. And so when we get there, we've actually just managed to get some really good trajectories. We are interested in looking at how they move. So they are very persistent beings, but they also have these types of run and tumble trajectories, which are very common in bacteria. But since they are ciliated, they have the capacity to go back and forth and do these crazy moves. And so we're looking at that right now. And for the first time, actually, like the week before I came here, we got some really nice data. We're looking at the times when they're very persistent. But, well, why? Why do we care about paramecia? Well, first of all, I think they're very cool. And I think understanding their movement can also be very cool because they're a little bit different from bacteria, which is where most of the data and literature focuses. But also, our end goal is to poison them. So why? Again, so we see with some animals that their behavior changes when they are in substrates that are bad for them. And paramecia, we've seen that when they have less oxygen, they start to move very slowly. And if we could poison them, let's say, and see that their behavior changes, that their movement patterns change, they could be really good bioindicators for water quality. And one thing about them is that they're very cheap to have in a lab. You only need like a little cup of them. You can throw them in the sink. And you can poison them because nobody actually cares what we do to protozoans. We don't have to deal with any ethics councils or anything. So if we find that they are good bioindicators, they could be really good alternatives to calculate water quality. So this is, a general idea because it's very different from the other topics in the school. But this is sort of what we're doing right now. So thank you. That's my group. You may know some of them. And yes, thank you.
Okay, questions? No? Okay. Okay, Susanna, please. Just kill this. Okay, you know which of these is your file? Okay. Oops. Let's see. Let's try again. Reparar? No, it's not working. It's coming? No, yeah, it's here. Okay, yeah, something is here. Yeah. You want to try to make another copy? We can move you towards the end. Yeah. Okay. Okay. So we have a problem with the presentation, so we're going to make another copy and see if it works. So we move to the next talk, Martin.
Thank you. Okay, so it's how can we?
Full screen.
Yeah, maybe. Yeah, perfect. And where is this button?
This one, yeah, this one.
Is that ChatGPT?
Maybe. Okay, so hi everyone. I'm Martin Guillemot. I'm a third year PhD student at the Paris-Bern Institute, and I'm working with Mario Chavez, who is present today. So the title of my flash talk is Mapping Brain Network in Hyperbolic Space, a game changer for epilepsy surgery forecasting. So a very catchy and sexy name with a sexy AI simulated figure just to get your attention for the next 5 minutes. So, as you may know, we are going to talk about epilepsy and epilepsy surgery. So maybe you know that about 1% of the population around the world is epileptic. And for 80% of the person, we can control the seizures with some drugs. But when it's not working and the seizures still continue, we can, for some people, use surgery. We will remove a part of the brain. For example, in this case, I will show you. And then the seizure will stop. However, in 20 to 30% of the cases, the surgery does not work, and people still have seizures, and we don't know why, and we can't predict it before the surgery. So the objective of our work, in this case, is to see if we can predict if the surgery will be a success or not, and to try to find markers of it, and maybe to find new region of the brain to remove to stop the seizures. To do that, we'll study the brain networks. So for each patient, we'll get the brain network before surgery with DTI and MRI, and the brain network after a surgery, and we want to compare them to see how the surgery affects the network. And the post-surgery network, we can simulate it by drawing into the MRI the region that we want to remove. For example, for this patient, this part of the brain has been removed. So you see here is before surgery, and after surgery, you have a wall, and you have here the post-surgery brain network. To compare it, we'll use hyperbolic geometry. So I think everybody knows in this room Euclidean geometry. That's basic, and everybody uses it. That's the case here. This piece of art, you see all these the same sizes, and according to the Euclidean metric, they are the same sizes. Okay. In our work, we don't use Euclidean geometry, but hyperbolic geometry. So here you have an example of an hyperbolic disk, which is a representation in 2 dimensions of an hyperbolic space. And you see the horses with different sizes, but according to the hyperbolic metric, they are all the same. Okay, so when you go towards the radius here, you reach the infinity. And it's very well, well adapted to embed network, especially network with hierarchical structure as brain network. So that's why we will use hyperbolic geometry. So basically what we do, we take the pre-surgery network of a patient, we embed it in the hyperbolic space, and we do the same with the post-surgery network, we embed it in the hyperbolic disk, and we will compare these 2 embeddings to see for each node their position, how they move. Okay, and we will quantify the effect of the surgery in the disk. And so for this patient, for example, this part of the disk is highly impacted by the surgery, and this part of the disk is less impacted by the surgery. And the question now is, can we differentiate the 2 groups of patients, those for whom the seizures stop, and those that still have seizures after surgery? And the answer is yes, of course. For example, on the left, you have a patient operated in the left hemisphere, and we see that when we focus in this part of the disk, we're able to differentiate the 2 groups of patients and to predict whether the surgery will be a success or not. And what is interesting is that when we go back into the brain space, we can identify a region of the brain implicated in the surgery success. And when you are operated in the left hemisphere, you have to look at the right hemisphere to see whether the surgery will work or not. And when you are operated in the right hemisphere, you have to look at the left hemisphere to see whether the surgery will work or not. And we have quite good results. However, we only have 50 patients, so it's not a lot, but it's still quite encouraging. So if you want to go to my Scholar and see my papers of our team, you can flash this code. And here it's some people of our team in Marseille last year. So thank you for your attention.
Susanna, you want to try again?
Good afternoon. My name is Susana Ochoa. I'm from Mexico, from the University of Morelos in Mexico. My advisors are Thomas Seligman from Universidad of Mexico, UNAM, and Marcus Mueller of the University of Morelos. I'm interested in complex network, dynamical systems, and I've been working in time series analysis. For my master, I work in financial system, and now I'm a PhD student, and my work is on EEG data and human sleep. So today I'm going to talk about our work, who is already published. The name is Narrow Band Sleep Stages Analysis, Eigenvalues and Eigenvectors of the Multiband Cross-Correlation. So, well, some of you must know or know that the normal sleep hypnogram looks like this. So there are different stages which the specialized characterized just by eye, looking the signals. So if it's slow and they have a special shape, they can say, oh, this is stage 4, and if it's rapid movement or waves, and you know, they will see which stage is. So in the night, we're going to have, if we have like a healthy sleep, we're going to go for deep sleep to REM, which means rapid eye movement. So, and this is going to happen like 4 or 5 times per night. Like we're going to have like 5 or 4 cycles of the night. There are many interesting things in this system because as we can see, we cannot go for stage 2 to REM without pass through stage 2. So it means that stage 2 is like a bridge between these 2 different stages with rapid waves and slow waves. Also, we interesting to know how these stages evolve, each one around across the different sleep cycles. But in this work, we only focus on characterize the sleep stages using this cross-correlation matrix. The data we have there is for 10 healthy young men without disorders, sleep disorders, and it's for the sleep lab in UNAM. So we're going to filter these signals. We're going to have 19 signals, and we're going to filter them in a bandwidth like is small, which is 2 hertz. So the border is going to be from 1 to 3, 3 to 5, and so on. How we build our multiband cross-correlation matrix? Well, from these signals of the electrode, we're going to have 19, as I said, and then we're going to filter it in 40 bands. So now we're going to have 19 paired 14 times each signal. So this 19 signal, we're going to filter in 1 to 3 hertz and so on. And we're going to take like a time window, which is going to be 30 seconds, which is the same that the specialized used to label the sleep stations. So then we're going to construct the correlation matrices using a Pearson coefficient. And then when we're going to move this window across the whole night. So one of our results is when we get the eigenvalues of this matrix, but we take the mean over the night, we will be able to match more with the scoring. In the image, we are seeing the evolution of the eigenvectors of this cross-correlated matrix. And this is the minimum eigenvector, the maximum eigenvector, this is the time. So this is a heat map, and the black line is the scoring that Specialized give us. So I don't know if you can see it, but for example, here we can see like the eigenvalues are matching with the scoring. So this is one of our results, like the relative eigenvalues, which mean these like normalized eigenvalues without the mean in the standard deviation, dividing the standard deviation. It's better to describe the different leaf stages. I don't know. Okay. So we also analyze the eigenvectors, but I don't know if I have time. But if you have doubts, you can go to my poster. And if you are interested in this, you can check the paper.
Thank you. Okay, so next is Malena Hidalgo.
Hi, thank you. Do you? Okay.
Yeah.
I'm changing slide.
Yeah, you can also use this, the arrows, and this is the laser.
The laser?
Yeah.
Okay. Okay. So, hello everyone. My name is Malena Hidalgo, and I'm an undergrad biomedical engineering student in Universidad Nacional de San Martín in Argentina, and I participated as a research fellow in the SEMC3 laboratory with under the supervision of Daniel Martin and Dante Chialvo. This is the top of my poster. If you can go and search for it later. So throughout the whole project, our main goal was to distinguish between patients with schizophrenia and control patients based on their brain region connections, generating simple simulations of their dynamics. So we started with structural connectomes that we got from Synodo. And with that connectome of 83 regions, we added, with a simple model that I'm going to explain later, we got the dynamics, artificial dynamics. With these dynamics, we got an analysis, and then we try to classify between both groups. So with this, we also, our project was we also oh, sorry, I'm nervous. We read a paper that did similar things, but with structural, functional, and the combination of both, with some machine learning tools. So we wanted, our first question was if we could do the same thing or even improve them with artificial dynamics. So we used the Greenberg-Hastings model. I don't know if you know this, but it's a simple model with discrete nodes that evolve in discrete time steps. So these nodes can be found in 1 out of 3 states that are active, inactive, and refractory. One node can go through one state to another due to a probability, or can go to an active state due to the sum of their neighboring connections. Due to a sum of the neighboring connections, if it's bigger, it's higher than the threshold, that it's the control parameter T. With this, we try to do it with 200,000 time steps, and we analyzed the first autocorrelation and found some critical dynamic close to the parameter T equal to 0.02. But we found interesting results in supercritical values of T's that were lower in this region. For the classification, we use these tools that are support vector machine and RFE, recursive feature elimination. And for 100 iterations, we divided the data in 90% so we can train the model and eliminate the features that weren't as important, and the other 10% so we can test the model's performance. So to test sorry. For the model's performance, we used the accuracy to evaluate it. And here you can see the number of features. And we found that with our artificial dynamics, that are the blue and the green one, we found better results in accuracy than for the original data. That was structural, functional, and the combination of both. We also found or concluded that this artificial dynamics is important to use because it could remove the noise from functional data where the patient can move or the heartbeat can make some noise in the data, so it could improve the results. If you have any other doubts, you can find me or contact me via email or go to the poster and I'll try to explain it better. That's it. Thanks for listening.
Okay, next is Paula Rodriguez.
Okay. Hi, everybody. My name is Paola Guin from Mexico. And, well, now I have been working at the Universidad Autónoma del Estado de Morelos as a junior research. Since the last year, I'm very new in this part, but now I want to talk about one part of my PhD and postdoctoral project. Well, the title of my talk is very large, but we have little keywords. The first is the stable correlation pattern, dynamics, modulation with age, and processing on phases in children. Well, I'm really interested to study the brain activity using the functional connectivity, in this case, using EEG data. We know that the EEG data record then the electrical activity of the brain, in this case, using electrodes in the scalp. All the people that try to understand the brain using EEG, we know that different things to consider. The first is these signals are contaminated by noise, not only in the noise of the system, if not the noise, because sometimes the patient moving or the people begin to blink, and these affected signals. And the second big problem is the volume condition. And the other, that there are a lot of literature or articles to explain this, is that the functional connectivity is modulated by the EEG reference. That is a big problem because we don't know what is exactly the best EEG reference that we can apply. But this is other project. Maybe if you have question about this, I can explain later. And the other big problem, we know that this EEG signal is highly non-stationarity. But the background of my project is this article from Marcus, and he studied the functional connectivity using the correlation matrix in epileptic patients. Here we calculate the correlation matrix using the sliding windows, and he found this structure. I forget the color bar, sorry, but the color red corresponds to the positive correlation, and the negative correlation corresponds to the blue scales. The electrodes here correspond to the left hemisphere, center electrodes, and the right hemisphere. And the principal idea is here we found a stable correlation, a stable positive correlation inside to the hemisphere, but between the hemisphere have anti-correlation. And the most surprising to this is the same when we are in the epileptic seizure, and pre-ictal seizure, and post-ictal. Maybe this is important because we know that the brain activity during a seizure is totally different. No? Then we have another things is very stable between patients. And maybe this stationary pattern that we call is a manifestation of the brain activity from epileptic patient. And the first question is, what happened with healthy subjects? Maybe we have other different structure or a correlation structure. Here we're using the same data that Elsa explained. Using the sleep data, we found the same structure that is very similar to the epileptic patient, and not it's only in the different sleep stages. It's very stable in different frequency range. Then we have one interpretation using a nonlinear model to try to explain why the brain have this structure. But now we have more questions than answers, but the poster is trying to answer this question, is what happened to this structure in the different age. We know that the brain changes with the age, and maybe this structure depends the age of the subject. And the second question is, what about the dynamics of the brain activity? We know that the brain changes when we are listening something, when we are thinking, but he always is stable. What happened, no? And we propose a new methodology. In summary, is this structure or this stable correlation pattern is a manifestation for this phase space we can to reconstruct, but the brain dynamic is equal to this structure plus fluctuation. These fluctuations are deviation around to this stable correlation pattern. In order to prove this theory, we apply this analysis to EEG data from children. In this case, we have 3 groups, the children of 6, 8, and 10 years old, and we try to explain if this structure is the same in the different age. And the other thing is what happened with this modulation of fluctuation around to this stable correlation during a resting state and during the task. The task is an old boy, I forget the name. The task is the kids try to recognize the face, a specific face, recognize if it's a woman or a man, or see the emotion or recognize a specific emotion. In this case, is happy, the emotion that the kids recognize. But if you look the picture, it's so difficult to try to separate this. So that's all. If you have more questions, I will try to answer. But the idea is we have always questions about this project, but that's all. Thank you. And
if you have quick questions, it's okay to ask questions. I mean, we have time.
As a function of the age, what do you learn? I mean, the kids that are much younger with respect to the ones that are 10 years old are able to do everybody the job of distinguishing?
Yes, it's really we try to answer this question. Yes, we all the subjects doing the same task, but the more surprising, the younger children, it's more difficult to do in the task. Specifically, when you try to recognize the sadness phase, that is more difficult for the younger, that the children that have 10 years old. That is, yeah.
Thank you. Okay, next, Jieru. I'm not sure I'm pronouncing correctly.
Thank you. Okay, thanks for the chance for sharing my project about measuring instability of brain networks in epilepsy. So in previous talks, we are already familiar with seizure. It's a sudden uncontrolled disease. And we can see in this EEG data, when the seizure happens, there will be fluctuation and it's rapidly spread to the whole brain. So a common way in this field is to analyze its propagation is to conceptualizing brain as a network model where the epileptogenic zone, EZ, is a node that drives perturbation to the other nodes. So what I want to investigate is to find the dynamic nature on epileptic network. So there's also a classical statement says seizure can be seen as a phase transition from a stable state to an unstable state. So maybe is there a feasible solution for instability as a measure on this? So previous measure to measure instability is using extreme eigenvalues, which it could somehow reflect the network dynamics and structural property of network. However, this measure is indirect since just two eigenvalues could not reflect the full properties of network structure, and it can't tell us about how this highly connected area is the main driver of the instability. So our goal here is to, based on the network structure, we want to develop a direct measure for instability so that we can answer this research question. So here are our new instability measure. We start describing the dynamics for how much a network is deviating from a stable fixed point. And then we link this deviation to a network structure. Specifically, it's by calculating all the convergent works in the network. And also, we develop node-based measure on the same principle. So now we can start answer our research question for representing seizure development. We use the epileptic model, which each node's dynamics described by this equation with the key parameter x0 means excitability. And we start from a healthy brain state, which each node, x0 is minus 2.3, and we choose a node as easy and increase its x0 to induce the seizure, so that we want to find the gradual change in this process. And this is our measure result. So our instability has identified the stable state and then a turning point to the unstable state. So here's the increasing instability measure. So the second research question is we want to use our measure to quantify node susceptibility. So first one, we calculate the critical x0 from the model as ground truth. And secondly, we calculate each node for their impact on the whole network's instability when the model is setting as a healthy state. So by mapping the figure 1 and figure 2, we can do a comparison for how well for our instability serves as a proxy for the epileptogenicity. So this is a result. It shows a very good match. It not only shows that instability could tell us for how possible for a node to trigger a seizure, but it also be measured in a healthy state, which means it could have some potential to do some predictive things. For example, it can be a pre-idle EEG marker, which is also our next step research. So here are some main takeaways of my research. But what I want to discuss with you is this question for the synchronization and instabilities, some subtle difference. And the second one is about how to better use this model. And then it's my next step research. I want to discuss with you how to apply it on the EEG data. So thanks for listening, and I'm happy to contact and talk to you.
EEG turned into a song? They went really quick.
You have music going on?
Okay. Okay, thank you. Okay, so let's talk of the days by Antonio Gabriel. He's going to do it on the blackboard.
Hello there. It's the first time I'm talking in English for a lot of people, so if you have any questions, you can talk to me directly. So I'm a applied mathematician. I'm an undergraduate student, but my research is about data modeling. I've seen a lot of your researches were incredible, and they have a thing in common: data collection, data treatment, data, everything about data. And I would like to show an approach of the treatment of this data. And okay, so we have a problem on data collection and data modeling, that is the mix of continuous and discrete data. As like sometimes you can have 0.9, 0.8, or 0.75 data, but sometimes you can have 1, 0, 0, 1, and in some cases it can be, oh my God, what I can do about it? We exclude this data or something. My advisor, Francisco Felipe, created a model which can, how can I say it? Which can treat this kind of data. And the first thing he created is a power logic model that his utility is for symmetries, this kind of data. So we have, as an example, a frequency of this structure. And with the power logic defined by this Z, we can have this kind of data more symmetrically. And each follows this structure, and this structure have this RFZ more than 0. It's called a density generator function. That is, you can choose a distribution that can serve for your amount of data. You can use, but the condition is that the distribution needs to be symmetric, as normal, student T, etc. More information you can find on some papers of Francisco Felipe. And the probability density function follows this structure. Lambda is a skewness parameter. The sigma is the dispersion parameter, and the mu is the centralizing parameter, like the median. And the thing I'm already developed is this distribution that follows a convex combination between a Bernoulli with gamma probability. That is, something happens or not, and the accumulated distribution of the power logic distribution. The next step of this is create the regression model for this. And the application we intend to use on this is that the shift and catch of tunas due to global warming. Many of these data have 100% or 0% on some tropical region, on some regions of Pacific Ocean, Indian Ocean. That is. And yeah, is that it? Thank you very much.
Thank you. Okay, so this is it for the presentations. Thank you very much for all the speakers. This was very nice session. So do we have any announcements to do? You want to say something about the next Thursday, about? Yeah. Okay, so you see, usually in these schools, you were presented with some homework problems, right? Yurik and who else? Carmen gave you a set of problems to work on. And usually what we do in these schools is that we really ask you to work on these problems. And if you get somewhere, if you manage to solve one of them, it doesn't have to be anything spectacular. Just do some work. You have the opportunity to present it to us next week. So next Thursday in the afternoon, we can do something like this, but not about your own work, but about what you learned here. Yes? You can do it in groups. Yes, you can organize yourself in groups. And you see, this is not really mandatory. We know that you have lots of classes, but it would be nice if you could really show that you are doing some work, right? You have all these afternoons free, so it would be nice to get something done. So yes.
I just wanted to say that I refined the slides with the exercises. And so because Amelia told me also that the paper